So I would like to call Professor Yael Mandel-Gutfreund, our newest professor at the Faculty of Biology, who is also a collaborator of Ada Yonath, to introduce our first speaker.
Thank you, Benny. Thank you, and it is a greatest honor and pleasure to introduce our first keynote of this meeting, 2009 Nobel Laureate Professor Ada Yonath. Professor Ada Yonath won this year's Nobel Prize in Chemistry for her studies on the structure and function of the ribosome. Specifically, Professor Yonath was recognized by the Nobel Committee for her pioneer work in crystallizing one of the largest molecular machines in a cell required for protein synthesis in all living organisms. For many years, visualizing the ribosome in near atomic resolution seemed to the scientific community, to be completely non-realistic and perhaps just a dream. But Ada made this dream true. Looking back at Ada's academic career, starting in the late 50s or beginning of the 60s as a bachelor's student in a Hebrew University in Jerusalem, Ada could surely be considered as a dreamer. Her ideas were always ahead of time, but her persistence made them plausible. In 1970, Ada Yonath established the first protein crystallography laboratory in Israel, which for nearly 10 years was the only such laboratory in Israel. During these years, Ada became more interested in how proteins are synthesized and specifically on the structure of the ribosome, the protein factory of the cell. In 1980, working on the halophilic bacteria ribosome, which she thought would be the most stable ribosome, Ada managed to obtain the first crystal of the large ribosomal subunit. This pioneer work, together with methods she developed to solve the phase problem, opened the door to her and other researchers to finally obtain, in the year of 2000, the high-resolution crystal structures of the two ribosomal subunits. Since the first crystal in 1980, Ada Yonath's group has solved the structures of the ribosome in complex with more than 20 different antibiotics. This ongoing work in her lab has contributed tremendously to our understanding of how can the peptide synthesis in bacteria be selectively stalled by the antibacterial compounds, which in many cases are produced by the bacteria themselves to fight each other. Having the detailed chemical interactions between the drug and the bacterial ribosome from the different crystal structures from Ada Yonath's group and others paved the way for designing new antibiotics for clinical use. This contribution is of great importance for fighting drug resistance, a major cause for millions of deaths per year over the world. During the last years, Ada Yonath has published inspiring work on the evolution of the ribosome. The recent discovery in her lab on the universal symmetric region in the catalytic core of the ribosome gives us the first glimpse to understand how the first peptide bonds were evolved in the prebiotic world. These series of discoveries and others I did not have the time to mention awarded Professor Ada Yonath over the years many important prizes prior to the Nobel Prize. Among them, I would like to mention the Harvey Prize awarded to Ada Yonath by the Technion in 2002 and the Israel Prize awarded to Professor Ada Yonath that same year. In addition, Professor Yonath is a member of the National Academy of Science, the American Academy of Arts and Sciences, the European Molecular Biology Organization, and others. And now, with no further delay, I'm delighted to invite Professor Ada Yonath to tell us what was first, the genetic code or its product. Thank you.
must say that I'm pretty embarrassed. Thank you very much, and I wanted to start in the way I'm starting now. Yesterday I came back from a meeting, actually in Amman, about the synchrotron, the Middle East synchrotron, and tomorrow I'm going to Rome. In between, I found time to come to this meeting. Benny convinced me, but something else convinced me too. And this is the support I got constantly from the Technion, although I was known as a dreamer. So even in 1990, just in the beginning, I got the Koltov Prize here at the Technion, and later on the Harvey Prize. The new president got together with me and met, and the old president pushed me all the time, further and further and further. And I feel in the Technion, although I never studied here and I never really did work here, I feel at home. And thank you very much for letting me express it today. So what was fair, the genetic code or its products? I heard that not everybody is a biologist here, so I take the liberty to spend 2 minutes of what is the product of the genetic code. These are proteins, and the story is that the DNA has the genetic code in it. It's being transcribed to messenger RNA, and it then being translated to proteins by the ribosome. So this is the part of the cycle that I will talk about today, and this is the product, of course, this is the code. So the code comes to the ribosome on messenger RNA, which is a long nucleic acid chain, which has somewhere in it a place where it has to start to be translated in triples. Each triple is a code for a special amino acid or specific amino acid, like that, for instance. The initial code to be translated is found and is put on the ribosome. I will show in a minute. And the translation is made good. I promise it was okay a minute ago. And it is okay here. So why not? And then that translation is mediated by another RNA molecule that makes base pairing, Watson-Crick base pairing on the messenger and carries the cognate amino acid at the other side of this helical L-shaped molecule. All the tRNAs in the world, from bacteria to dinosaurs probably, are of the same L-shaped structure. They may have differences in their sequences here, but they always have an anticodon loop here and a universal CCA, three nucleotides to which the amino acid is built. All of this, as I said earlier, is happening on the ribosome. The decoding on the small subunit and the peptide bond formation on the large one. So from the side, it looks like that. Initiation complex is made by messenger wrapping around the narrowest part of the small subunit. The first site to be occupied is called P site. So on the ribosome, there are 3 sites, 3 subsites, A, P, and E, where the tRNA will come. Here, this blue thing represents a tRNA with the amino acid. P is peptidyl transferase. It means where, at the end, protein will be made. A is the site where the new tRNA will come as amino acid, amino-acylated tRNA, and E is exit, as always. When this complex is made, of course, with initiation factors and other non-ribosomal factors that I don't show here, the large subunit can come and make quite nice contour recognition contact. The P site tRNA is now standing on top of a tunnel that spans the large subunit through which the newly protein will exit out, will emerge out. Bridges are being made between the two subunits to make sure that the association is completely precise. These are these bridges, and they are made by conformational changes mainly on the large subunit. And there are 13 of them. I show here only 3. The most important one is this one that actually leads from the neighborhood of peptide bond formation to the neighborhood of the decoding site, the two most important functional sites of the ribosome. Then reaction can start. A site can be occupied. It will later on go to here, and a peptide bond will be made. Now the blue is already empty, can go out, and a new A can come in. Sequence of events here is actually much faster than what I'm doing, and the A site is being occupied while the E site is, tRNA is leaving. These little arrows are showing the direction of the step in protein biosynthesis that is called translocation. So there is translocation of A into P into E, and a new A can come, and the protein can grow. So for children, when I give lectures to children, I show the ribosome as a factory that gets the instructions by messenger RNA. Many tracks all look the same, or more or less the same, bring the amino acids. The protein comes out here, and the tracks come out there. And I really love this little slide, and many adults told me that only after I showed it they understood, so I'm showing it here too. And according to our crystallographic studies that Yael already discussed earlier, we made a movie back in 2000 that showed what we understood crystallographically about the function. But I must put the light off for that. I'll try to do that. There are some achievements in life. And usually the biggest one I see, except for giving lectures to very, very young kids, is to have control on the lighting of the lecture halls. Okay, so the messenger is now coming to the small subunit and wraps around it. And pay attention now how it takes it. Choop, you saw? It moved. Shall I start again, or it's okay? Now the first tRNA can come. All these blue things are factors, and I won't talk about them, but of course the ribosome doesn't work alone. Now they can come out, and the large subunit can come and make the bridges. And actually now we have an active ribosome. So we'll have a look from the side. tRNAs are here, messenger is wrapped there, and protein will be made in the large subunit above this tunnel that is now being somehow highlighted. The ribosome helps the tRNAs to come in according to the genetic code and to come out. Now we'll take away the large subunit and look only at the peptide bond formation region, where the translocation happens sideways, but something else is happening down there. It's not exactly sideways. Things are happening here, and actually this will be the topic of the lecture. The protein comes out through the tunnel. Now you can see the whole ribosome. It will come out here. tRNAs come in and come out with amino acid and empty until stop codon is realized. And at that point, release factors and recycling factors are replacing the tRNA, and the two subunits dissociate, ready for the next protein. So that's the end of the story. The movie was done by two really nice students from Bezalel, Eyal and Zohar, and they got their MSc for it, and we were really working together with them happily. There are many ribosomes in each cell, and they are working very fast. Each living cell has thousands of ribosomes. Even bacteria can have something like 100,000 in the log period. In normal human cells that produce themselves, there are about 1 million in each, and 6 million can reach, the number 6 million can reach for the ribosomes in the liver cells. They're working very fast. They make about 15 peptide bonds in a second. For the chemists here, I'm sure neither of you can do this. Actually, I can't. Not even one in a second. 15 to 20 in a second, so a whole protein can be made in a few seconds. And the mistake rate is very, very low, 10 to the minus 4. The ribosome also has a proofreading machinery that even this mistake rate sometimes is caught by the ribosome itself and taken care of. So the reason that they can work so fast is shown here in a very old, very, very old slide from the 70s. It's an electron micrograph where you can see ribosomes, many, many ribosomes, translating the same messenger RNA. So it's low resolution and it's low contrast, but it's a wonderful slide in my opinion. The proteins come out of the ribosomes here as extended structures. Of course, because it was done on a EM grid, it's not like this in the cell, but otherwise it really shows. You see many, many ribosomes translate together. You can even see the two subunits, for instance here, small and large. Protein comes out of the other side where the messenger is being read. Actually, everything was known before us in the 70s. What do we know more now? So one thing I already showed you, the cycle, and I also want to show you that the ribosomes really have structure. They are not just these fuzzy bodies I showed earlier. They are made of, all ribosomes are made of RNA and proteins. In all ribosomes, except for mitochondria, the RNA takes care of 2/3 of the mass. So in the small subunit, what you see now rotating, which is called 30S because of sedimentation coefficient, there is about one RNA chain with about 1,600 nucleotides and about 20 to 21 proteins depending on the source. But in each cell, it's the same number. Actually, what you see here is a bacterial, eubacterial ribosome. And these are the only known structures today are of bacteria, archaea and bacteria. There is still not any structure of higher organism. The tasks of the small subunit, as I said before, they are decoding and also proofreading, fidelity control. And the large subunit, which is larger, it's about 1.5 million in molecular weight. It has 2 RNA chains, 3,000 altogether. About most of them are in a very large one called 23S RNA. I will talk about it later. And up to 34 different proteins in bacteria. Again, 2/3 is RNA. The RNA, I've forgotten to say, sorry, is here. The silver also was in here. The silver part and each protein in a different color. Colors were completely randomly taken. There is no meaning to the colors. So I just want to show how great this machine is and how complicated it by focusing on some dynamics. So the dynamic parts that we saw in the large subunit are the 2 hands that you saw in the movie, up, 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 the 2 stalks, and I show here 2 bridges. This is the important one near the active site. And in the small subunit, in the large subunit, first, one of the hands, for instance, this one, it's the exit. The exit stalk is made of almost 100 nucleotides, which is closed and opened either in the whole ribosome, that is called 70S, the 2 subunits together, or in the unbound large subunit. They both hinge around this point, chick, chick, every 15 times a second, this huge, huge part of 100 nucleotides and a big, relatively big protein, 20,000 for ribosome, it's the biggest. And this opening, closing can be iterated from 2 crystal forms, one closed, one open, unbound and bound, and I docked here the free tRNAs in order that you can see that this is closed door and this is open door. That's the hand movement in the movie, in the like bare ribosome. I want also to show a bridge formation. Here is a bridge, 869, that goes from the more or less the decoding site up here to the peptide bond site down there. I here represent the large subunit on this side just by this what will become a bridge in a minute, helix 69 it's called, and the small subunit will be here. And it will be represented by the initiation pieces, which is the initiation more or less complex, the P-site tRNA bound to the position where decoding happens. I don't show the messenger because it's just too messy. So here they come. The decoding site helix in the small subunit and the tRNA bound to it. Now this is the way the tRNA is bound in the whole ribosome, and of course there is here a conflict in positioning, in space, available space. So a tRNA bound, when it bounds to the messenger here in the small subunit, is much more rigid and much more strong than the end of this helix, which is very flexible. So there is a motion that makes a bridge. Here you see, it was like this, and now there is a bridge, a wonderful bridge. So that's the way bridges are made. And I show this just to show how much dynamics is within the ribosome when it functions. But I want to concentrate on a different region, and this is a part of the large subunit, two-dimensional representation of just the RNA, the longest RNA chain, 23S RNA. And you can have a look at it. You also looked at the ribosome earlier. I could not see any symmetrical region here, sub-region symmetrical. Also, when one looks at the sequence, there is no clue for any internal symmetry within the ribosomal RNA. But Ilana Agmon, that I saw in the morning, and I think she's sitting here, and she is a Technion product, she found it. She found that this part of the 23S RNA has internal symmetry, which is expressed only in the fold of the RNA. This region contains this internal symmetry, non-real symmetry, biological symmetry, and part of it, this part, relates to this part by a matrix of about 180 degrees rotation around the imaginary axis. Now, when I say around 180, what Ilana found is 179.5, and I hope that you agree that we can round it to about 180. So let's look at it in a little bit more detail. This is one part, the top part and the lower part of the region. This region, the lower part, it contains the loop where tRNA binds to the A site, so we call it A region, containing the A loop. And this region contains the P loop, where the P site tRNA binds, so we call it the P region. And I'll keep blue and green throughout the lecture. The two nucleotides here that are actually confining the one region from the other, like the imaginary twofold rotation axis, are the two nucleotides that are at the upper top of the tunnel from where the protein will go. The region has about 180 nucleotides, which is quite large. tRNA is 76. And as I said, it's mainly by the fold and also by the orientation of the nucleotides. It exists in all known structures today. What we have here is only 5 of them. Deinococcus radiodurans, this is a very good time for me to tell who did what. Deinococcus radiodurans, the large subunit made by us. Haloarcula maris morti, the large subunit determined at Yale. E. coli whole ribosome, empty whole ribosome, determined at Berkeley. And two structures of the whole ribosome with some of the substrates determined at MRC in England and also at Santa Cruz. You can see that they can superpose totally one on top of the other, almost fully, with the twofold axis coming towards you. the imaginary one. And I want to tell you, Yael already said that there are more than 20 structures today with antibiotics or with substrate. Actually, there are more than 30 structures, and they all can be superposed. I just didn't put them in this slide. So now if we look at it a little bit more, first of all, for those that didn't see enough symmetry before, we overlap the blue and the green around this axis, this imaginary axis, and you can see the amount of overlap. I hope for biologists it's very convincing. And you can even look at part of it, one on top of the other. But what I want really to look at is the active site. So what you see here is this fold, the RNA fold around the active site, with A site in here and P site in there. And I hope that you can see some remnants of symmetry even in this two-dimensional projection of quite a large three-dimensional volume. I'll put now the nucleotides. And as I said, there is no sequence correlation at all, but there is orientation correlation. And what we did, we colored the pairs identically. So this is this one and that one. This is a pair. This is a pair. This is a pair. This is a pair. You can see the amount of symmetry here. There is one nucleotide that bulges into the center, and I'll talk about it a little later. The A, as I say, the A site is here. The P site is there. I'll put now the nucleotide numbers for those that want to know them, and where the A and P end of A and P tRNAs. The CCA amino acid or peptidyl end of the tRNA meets the active site with the two-fold axis. So I hope that you can see that there is here a frame that the ribosome provides for the substrate. Question is, does the substrate take advantage of it? So for this, we made a mimic of this part of the substrate, the part that binds to the large subunit. This is tRNA, and this binds to the large subunit with the amino acid here that will come at the end to there in the yellow star. Now, please have a look at the mimic itself. The green star shows where the mimic stops. This is the part that should bind to the large subunit with CCA amino acid at the end. 35 nucleotides, and they bind very beautifully into the cavity that hosts the active site. The cavity is made mainly of RNA. There is one protein here, but it's really far from where peptide bond is being made. I didn't put numbers. You can easily find them, or I can tell them later. So the substrate that we call ASM, acceptor stem mimic, binds here and makes lots of contacts along the cavity. And here, this is the active site. Here, peptide bond will be made, actually here. So when we looked in detail how the substrate is bound, we found that the bond that connects the CCA amino acid to the rest of the RNA, it means it connects the piece here that is single strand to the whole double helical molecule. We found that this overlaps, more or less overlaps the imaginary two-fold axis, which means in our eyes that the tRNA motion from A to P is made of two independent, albeit correlated motions, a shift of the helical part and rotation of the 3' end. And in the movie, I try to draw your attention to this. Or if you want to look at it this way, this is more or less the diagonal way that shows the translation, the linear translation. And here, this part rotates around here. And if we look at it in a bit more detail, the rotation is here, and it is confined by the ribosome. These ribs are ribosome. So the ribosome makes sure that this is all what happens, and I want to show you a very nice real, it's not anymore a cartoon, but real. The volume that will take from blue to green, it means from A to P, is shown here. This is the occupied volume for this motion. The silver is the ribosome. So I think that you can see that not much can happen to the CCAA end, and here amino acids will meet each other and make a peptide bond behind there. From top, it looks like this, ribosome again in silver, and the volume, this will be the volume with the nucleotide that bulges in. And you can see that this is the only volume that is available for anything, nothing else. The ribosome is very, very dense. Otherwise, the rotation will take this direction. From the tunnel into the active site, it looks like this. Again, you can see how well the ribosome confines it with the two nucleotides. Actually, there is one underneath it, underneath the one we saw, and now it looks above because we look from lower part. They look like anchors or maybe do some other things. We'll discuss in a second. And the rotation will be in this direction. So when the rotation happens, you can see here, the part of the ribosome that is now shown in red that interacts with the rotating tRNA from A, tick, 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 to P, are lit. And the parts of the wall of the rotating motion is made partially of bases and partially of backbone. And this was nicely confirmed by comprehensive mutagenesis that showed that those bases that are needed for the rotation, that are interacting with the rotation, are essential, totally essential. The other have to be there but can be replaced. Also, we found that when the rotation happens, the transition state can be formed while the rotation goes on. And when we looked at this was found together with Massa and Carle by quantum mechanics. And when we looked at what can stabilize this, we found that those contacts that are made in the rotation are peaking exactly when the transition state is being made. So this is a very nice idea because A is already here when there is transition state creation, and a new A can come in, while this will be pushed out and protein synthesis can continue. What happens in the middle to the nucleotide that helps it is here just collection of many structures, and you can see that maybe this nucleotide also helps in the rotation, not only anchors it. At the end, the stereochemistry of the two parts of the tRNA, the A site and the P site, is perfectly made for peptide bond formation, and actually nothing else is needed. And recently, the structures came out showing the tRNA actually in structure in the active site. We superposed it on ours, derived one, the derived by rotation, the green, and you can see beautiful overlap, except that we had also, in our mathematics, we also put the substrate, and in the structures, there was no. So actually, this region allows for the two catalytic tasks of the ribosome. Ribosome doesn't only create peptide bonds. It also elongates them. It has to the next one, and the next one, and the next one. This is called elongation, and this is what the ribosome does. So what is needed for this, for the continuation? In terms of geometry, it needs that A site will be correctly placed. It means it will rotate correctly, and P site will go directly to its place in the beginning. Now, I would have a problem if I was a P site tRNA, going into an empty active site that is symmetrical. I have place here, I have place there. How can I can choose where to be? And if you think that every tRNA, first tRNA, has time to start to think where he wants to be, we won't have proteins and we won't be alive. So the ribosomes make sure that this happens correctly by providing here two candidates to anchor the P site. There is only one candidate to anchor the A site. These are both all guanines, G and GG, that can make boson pairs with C. So this explains also not only that the first CCA goes here, but also why CCA is so well preserved in all RNAs. And it was found also that when all these base pairs are being formed, the tRNA can be the catalyst of the reaction. It means the substrate is the catalyst of its own reaction, which is a very, very old, ancient way of catalysis, hardly exists in the modern life. So this now gives me the hook to go into how old is the ribosome. So what we studied until now, I actually said, it gives the frame and also allows for elongation and for substrate-mediated kinetics. I just want to show again this is the region in the large subunit. It is connected directly to the two arms that are moving when the tRNA comes in and out. You can see here the connections in red. And by the bridge in yellow to the small subunit, it takes part of all this region within the large subunit. And it seems to be able to transfer messages from here to here and from here to the messenger RNA, something that is very needed and was not thought until recently, because these are very, very remote locations. So you can see it also here if you didn't see it well before. Again, the blue and the green and the two hands with two tRNAs. So we think that the symmetrical region can transmit signals between functionally relevant, very remote locations, which is needed. It means that the messenger RNA has to know to translate by free nucleotides only after peptide bond is being made. How will they know it if there is nobody to transfer this information? So just have a look at it. I drew it here and colored it. I filled it up, and this is what we have, blue and green, and the entrance into the tunnel here. And you can see from here also how it looks from top, from the side, and how much it takes from the whole ribosome. That's the symmetrical region. Why did I speak about it so much, and what else do I have to tell about it? So I really have to tell about it something extremely important. It is very highly conserved. The ribosome is conserved very highly anyway, although mammalian ribosomes or higher organism ribosomes are much larger than the prokaryotic ones. They can reach 4 million instead of 2.5 million molecular weight, but it's mainly in the periphery. The internal part is extremely, extremely well conserved, and actually all ribosomes are completely universal. Each ribosome will translate any messenger you want. Doesn't matter what is the source. So with this a huge amount of very impressive amount of conservation, we thought that it actually indicates that its existence is beyond environmental conditions, or that what it suggests, that the protoribosome, which was a simple dimeric RNA enzyme, is still embedded in the core of the contemporary ribosome. And I want to show again, you saw it many times, but this is what I want to show now. This is the region with a little substrate here, embedded in what we see today as a ribosome in bacteria or in human or whatever. But first of all, could RNA exist as a machine in the RNA world? And recently there were several papers that support it, except for the whole idea of RNA world. It was shown that Sutherland showed in May, I think, no, it was May, that he can make bases by very, very simple chemistry. Shostak went out of his mind and immediately wrote news and views supporting it. And Joyce, a few months earlier, showed that RNA can replicate itself. So actually, RNA could be what we call today, could be a life, I don't want to say life molecule, but maybe a life machine. So our hypothesis is that there were pieces of RNA that could come together and make a structure that has a pocket in it. You can see it from top or from the side. And this pocket was able or made for accommodating substrates like CCA amino acid or any other amino acid, activated amino acid, come out by, if you have two of them, with a dipeptide and so on. The dipeptide can be now the substrate for the next one, and next one, the next one. So this is what our hypothesis was. And we suggest then that the chemical prebiotic process originated from an oligonucleotide and proceeds via self-folded unit into a self-assembled dimer, thus producing protoribosome pocket. That's a pocket I sort of showed before. And as RNA can be its own template for implication, the protoribosome could have evolved by gene fusion or gene duplication, which we think gene means RNA genes for itself, not the genes that we have today. So we looked into lots of structures, including the symmetrical region and other what is called very ancient RNA molecules, and we found that many of them have the same motif: stem, elbow, stem. If this is a motif that repeats itself even in the active site, perhaps this was the motif that we started with. So this is now the way we draw it. This is the motif: stem, elbow, stem. What is drawn here is just a primitive version for our construct. And this should fold to something like this, what we call today, and it's very feasible. It is even very feasible when one tries to, one tries to calculate what will be the three-dimensional structure of that using calculating programs. So this is one way that we try to construct this protoribosome. We don't only talk about it, we want to make it, to produce it. And the other way is to take parts of the ribosome as it is today, of the RNA as it is today, and build a protoribosome accordingly. And God, something really bad happened today. Oops. I'm sorry about this. But you can see here, you hardly can see on this side, please ignore it. I think that something moved here. But you can see here the parts that can make stem, elbow, stem, stem, elbow, stem, and the loops that are actually quite far, the loops that are accommodating the CCA upper top. So these are, for instance, other constructs that we made according to what you saw earlier, which have the sequence, which is very, very highly conserved anyway, but we are taking Thermos thermophilus as our starting point. The sequence that you see in the contemporary ribosome, with places here that have to be glued, inserted, because in the contemporary ribosome they have extensions. And in these places we also make what we call test tube mutations. You can see the real result in a poster that Ken Davidovich is showing later, and I only want to talk about the conclusions. So again, this is our hypothesis. Two parts are coming together, and I'm sorry that it jumps so much, makes it pocket, and this pocket from top looks like this, can accommodate two amino acids. What we change, as I said earlier, is the sequence and also the interface points here and here. In this we have control, and we can see what is the tendency to dimerize by changing this. The co-evolution of this proto machine together with the substrate is assumed to be incorporating this loop that is really far away in the contemporary ribosome into it in order to accommodate larger substrates. And for this, we try to make a construct that includes this loop. The loop is here in the stem, elbow stem, and we incorporated it into our construct. And we found that it doesn't prevent dimerization. Whatever P loop, P site dimerized, and with or without the P loop, the dimerization continues or exists. which shows that we still don't know whether the P loop was right in the proto-ribosome or came in together with its evolution. So what we really say now, together with the shadow of the contemporary ribosome, is that we had a pocket that could have been connected together between the A and P. This is the pocket. Whatever happened, this connection had to disappear, and the P loop could come then or be there from the beginning. And until now, I showed that this machine as lines, but it actually has volume. Here are the lines, the two backbones of the two sides of the active site. Here are the nucleotides and the volume that it takes, and a substrate can just come and fit itself in. So what could be the substrate in the old world? Just amino acid is not a good substrate. It hardly will make a peptide bond. It has to be activated amino acid. Did activated amino acid exist? We can see in this literature since the 1995 that tRNA can charge itself with amino acids. So the CCA and could have been an ancient substrate. So from the proto to the modern ribosome, there were genetically optimization, and we can show here. The backbone is still the same, but the bases point to different directions in order to adjust themselves to the different functions of the A and the P site. We got a lot of encouragement by a paper that was published in the beginning of the year from Steinberg, from Montreal, that says the same. The ribosome grew around the active site, and the active site was symmetrical site, and also from Heisor that says that the ribosome is a fossil. So what are the take-home lessons? Analysis of the early steps of protein biosynthesis enabled the visualization of a continuous path from the primordial world into contemporary ribosome translation. And it also indicated that ribosome is a naturally occurring ribozyme that outlived the transition from prebiotic RNA world to contemporary life. If you paid attention, I said it, but I didn't emphasize. The P subregion is preferred for what we found as dimerization. We never got dimerization of only A. Is it indicative or occasional? Does it show that the early machine was P site? It means a dimer of P site. The take-home lesson 2 is that the non-uniform tendency to dimerize, it means the preference of P site, was also already existing in the very old life, although fitness and natural selection is used usually for organisms, we think it was also there for molecules. And open questions. Was there an RNA weight at all? Our suggestions are also valid for PNA weight, but something had to be, some type of nucleic work. Was there a proto-ribosome and an RNA machine performing RNA needs prior to appearance of amino acids? That's a good question, because why the hell RNA will start to make proteins when it was so happy on its own? Did the proto-ribosome exist within a vesicle? We don't know this. And what was first, the genetic code or the product? This is the name of the title of this lecture, and I don't have an answer, except for suggesting that those proteins that could either perform better or stabilize the machine led to the emergence of the genetic code. Because this computer gave me so many problems, I will just come to the very, very last one. And this is the group that did the work in Israel, did the work that I talked about today, the identification and the construction of the proto-ribosome. Ilana is shown here. Ilana Agmon, that I mentioned, Chen, that will show the poster, and Matt are making the proto-ribosome. Anat, who is not here, is running the group. Tamara, our biochemist, biologist, had a birthday the day I took the picture, and look what she really likes as a cake. Thank you. Pick the prebiotic ribosome. And we will tell you when we have it. But the evidence is actually from the structure of today. What we think that today the structure has this machine in it, and this is why most of the lecture was to explain what the machine was, in order to convince you that we see a machine. What was, as I asked in the last slide, what could be the reason for RNA to make a protein machine, which I didn't answer, because I thought that maybe you want to discuss it. We don't think that it made a protein-making machine. We don't think that the RNA one day decided we need also proteins. We think that there was a machine for RNA, for something in the RNA, well, that RNA needed, and the amino acids, being so clever, pushed themselves in. And then when proteins became better than the RNA in many catalytic and stabilization functions, they took over. That's what we really think. And so in parallel to making the construct, we also look for which RNA duties it can fit. Okay?
Arye?
It seems like the proteins are folding as they come out of the ribosome because they have plenty of time to do that, if I'm not mistaken, or starting to fold. Is there any special properties of the surface of the ribosome that might suggest directing the protein folding during that process, or beginning of directing the protein folding during that process?
So as I had only 1 hour, and most of the hour I saw this yellow and red flags telling me finish, I didn't talk about it at all. Usually there are, in cells, there are chaperones that stick to the ribosome and help the folding or take the protein to a folding place and whatever. There are some evidence that some hydrophobic proteins start the folding within the tunnel, and about this there is so much dispute, even more than about the proto-ribosome. And in the test tube, they fold, but they take a lot of time by the sequence that is dictated in the gene. But in the cell, there are chaperones. Make sure that it happens, and correctly.
Yes, Doron?
Ada? In your movie, beautiful movie, the tRNAs are standing in line, waiting to enter orderly where they should. In fact, there should be a lot of impinging and departure for the recognition of the appropriate anticodon. Is there anything in the ribosome structure that allows that?
Okay, so that's a very great open problem that I'm not stopping thinking about it. And if I'm allowed to cite Aaron Klug, the Nobel Prize, Aaron Klug, he said, they stand in line like in England, not like in Beersheba. It means, because you know he has some contacts with Ben Gurion, it means that what he thinks, and what I also think, that there is some type of information given to them how to stand in line correctly and not push them away and come out. From space point of view, there is no way that a bad RNA can come in and be kicked out from the ribosome itself, except for stopping the synthesis. It has to be selected correctly before that. Ribosome, once they are in this very long mRNA-bound ribosomes, these polysomes, what we call, there is no space for the tRNA to come out, except for going into the elongation step. But maybe there is suggestion that the messenger already carries some type of signaling to the tRNAs to stand in line correctly. And I know about 5 or 6 very important scientists, very, very good groups that tried to look for this signaling and didn't find it yet. I'm pretty sure something will be found, and Aaron Klug will be right.
Okay. I think we have to continue, so thank you very much, Ada.